Sunday Night Science.
But first tonight, there aren't many areas of science that are quite as controversial as genetic modification. There's just something about the idea of making changes to the DNA of a living creature that we find a little uncomfortable, particularly when it comes to humans. So how do you balance safety and morality with all the benefits the technology can bring, like the potential to cure deadly diseases such as HIV, sickle cell disease, maybe even cancers? Well, that's something the World Health Organization is wrestling with right now. Just last week, they released new guidelines for how we should be approaching genetic modification, or as it is more precisely known, human genome editing. Well, to help us understand the complexities, I'm joined by Dr. Jo Milton, evolutionary biologist and science communicator at the Australian Science Media Centre. Welcome back to Nightlife, Jo.
Hi, Indira. Great to be here.
Yeah, great to have you. Now, this report from the World Health Organization looks at genome editing in humans. What exactly are we talking about here, Jo?
So, Indira, this is a sort of form of genetic modification known as gene editing. And basically, gene editing allows scientists to, well, literally edit our DNA. It's like a pair of genetic scissors, almost, which can be used to snip out bits of DNA. And then scientists can remove, add, or replace the DNA where it was cut.
All right. And there are different kinds of genome editing, aren't there?
That's right. So there's a few different methods, but they've really all been superseded more recently by a technique that's known as CRISPR-Cas9 that was invented in 2009. But perhaps the really important distinction in genome editing to be aware of is between editing what are called somatic cells and editing what are called germline or sex cells. Basically, somatic refers to making changes in regular body cells. Most of our cells are somatic, our skin cells, our hair cells, the cells of our organs. And when we change those, they can't be passed down to our children. But germline changes are heritable because they affect the genetic material that's in sperm or egg cells. So that means the intended change, as well as any mistakes, will be passed down to the next generation and the next generation after that, and so on.
Right, so these are the distinctive differences, somatic and germline. Very, very interesting. So, Jo, the way the science is portrayed in movies often makes these techniques seem very easy, that you just mix a little bit in a little bit and add it to a test tube and voila, you have a new gene sequence. But obviously the reality is very different. But genome editing has recently become a lot more accessible, hasn't it?
That's right, and it's all to do with a technology called CRISPR-Cas9, and it's a simple but very powerful tool for editing DNA. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which I think we can all agree is a bit of a mouthful.
Yeah.
And Cas9 stands for CRISPR-associated protein 9. So it was adapted from a defense system that bacteria have. And basically, CRISPR is a targeting tool. So it looks for and latches onto specific bits of genetic code. Then the Cas9 comes in because it's what we call an enzyme, and it cuts DNA. So wherever the CRISPR lands, the Cas9 comes along and acts like that pair of scissors to cut the DNA at specific places. So by targeting the CRISPRs to both ends of a gene that we don't like, that we want to remove, one that's causing problems, we can effectively cut out that problematic gene, and it can then be replaced with a healthy version. And hopefully that means the individual no longer suffers from the symptoms of that disease.
Okay, Jo. So where specifically is this technology being used in humans to treat what sort of conditions?
So it's really 2020 when we started to see CRISPR coming out of the lab and those kind of lab-based experiments and actually being used in the clinic. Early in the year, some US scientists said they'd used that technology inside the body for the first time. And they were treating an inherited form of blindness, which is called Leber congenital amaurosis. The doctors dripped fluid containing the gene editing machinery under the patient's retina in the hope that the problematic gene that causes the condition could be snipped out of the patient's own cells. So that was really one of the first major kind of clinical uses. Then later in that same year, some scientists from the US and Germany published a study. They'd used gene editing technology, the same thing, CRISPR-Cas9, to treat a pair of really nasty inherited diseases that interfere with the ability of the blood to transport oxygen around the body properly. And these diseases were beta thalassemia and sickle cell disease. So that was a study of just 2 patients, one with each disease, but the results were really, really promising. And then this year, there was a new development. We actually saw gene editing used inside the bloodstream for the first time, because previously cells had been removed from the body, edited, and then reinserted. But in this case, they injected the CRISPR-Cas9 mix straight into the people's bloodstream, and they used it to treat 6 people with a nasty, potentially fatal condition called transthyretin amyloidosis. They received the therapy, and they all showed a drop in their bodies of the levels of this nasty misshapen protein associated with the disease that's caused by the faulty gene.
Right.
So that was really very promising.
So really important breakthroughs you're describing there, Joe. Why then is the World Health Organization now getting involved?
Well, Indira, it was really prompted by a huge scandal in 2018, and it really sent shockwaves ripping through the scientific community. It was a Chinese researcher called He Jiankui, and he announced at a conference that he'd actually created the world's first gene-edited humans, so the first genetically modified babies. He said he'd used CRISPR to edit genes in 2 embryos, and both of those embryos had already been born. They are babies called Lulu and Nana. Now, the aim of his gene editing was to make these babies immune to HIV because their father had HIV. And he said that in one of these twins, 2 copies of the edited gene had taken, and that baby should therefore be immune to HIV. But the big thing here is that these are germline changes, so it's not somatic. And that means if these babies go on to have babies, they will inherit those altered genes. The world was not ready for this, and it sparked absolute outrage among the scientific community. And none of it could actually be verified because it was announced at a conference in a sort of press conference situation. Nobody has actually seen his work, and nobody has actually seen Lulu or Nana as well. So none of it could actually be verified. But China, in response to this international outrage, stopped his research immediately. And he has actually since been found guilty of conducting illegal medical practices and sentenced to 3 years in prison. It was also revealed that he and 2 of his collaborators had forged ethical review documents, and they'd actually misled the doctors who implanted those gene-edited embryos. They didn't know what they were doing. So that whole scandal led to the establishment of the committee behind these new recommendations and guidelines. Right. The WHO is a fairly slow-moving beast, which is why we're just seeing them now a few years later. But to be fair, it was a big process, involved hundreds of different participants, and they tried to bring in the views of all sorts of different groups, not just scientists and researchers, but faith leaders, indigenous people, patient groups, that kind of thing. Okay. So it's really the result of that big scandal.
So, Joe, does that mean that all germline genome editing is currently illegal?
Well, it is in most countries. Now, the WHO guidelines here call for a ban, basically, on heritable gene editing at the moment. But that's not all that it's about. It's got some other intentions as well. And some of the more important or interesting of those intentions are to try and ensure that new developments benefit everyone and not just the very wealthy. There's also some, they're looking to establish a registry of gene editing trials, so everything will be registered. They're trying to encourage international cooperation and to try and deal with those kind of rogue countries or bad actors who might not, pay any attention to these guidelines. They're also trying to set up a system to help whistleblowers to report dodgy experiments or practices happening around the world. Another thing that they're interested in is intellectual property, that kind of spectre of parts of our genome being patented and setting ethical standards.
So this case with the HIV babies came out of China. Is that where more of these instances are happening or potentially are, or are we aware of other rogue scientists in other countries as well?
Well, we are not at the moment, and China was pretty embarrassed by the whole thing, and it was not aware of what was going on, as far as we know. All these documents and ethical considerations, things had been forged, people were lied to.
Right.
So this stuff could still be happening. There are countries where the laws are played a bit more fast and loose than others, and I guess that's the concern. However, what the WHO says and does, none of it is legally binding. They're not the world police. So questions have been raised to, whether they have really got the power or if they're just very easily ignored by countries who choose to plough on with this kind of research.
Well, I want to bring in Professor Merlin Crossley now. He's a professor of molecular biology at the University of New South Wales and actually uses genome editing technology in his work researching inherited blood disorders. Welcome to Nightlife, Merlin.
Hello, Indira. Hi, Joe.
Merlin, what's your take on the WHO report and what impact is it going to have on your research in this field?
So I'm very pleased to see the report. I'm pleased for the guidance that is out there, and I think will have a beneficial effect on curbing any rogue elements that may have some ideas like He Xuankui had. It won't have any effect on my research. In Australia, research is highly regulated. There's no research that I'm aware of anyone trying to do germline gene editing, but many people are doing editing in the lab on cells with the idea, as Joe was talking about, providing therapies for patients who are suffering from diseases and somatic cells so that it wouldn't go through the germline. It would just be a treatment for the life of that patient, a sort of genetic surgery for the life of that patient.
And as you said, Merlin, you're not aware of any of that germline editing going on, but you personally as a researcher in this general field, what are your main concerns about that inherited germline style of editing?
So I think the absolute threat of this, if you look back to the days of cloning or the early days of stem cell therapy, it's not as if, there have been reports of various things happening here or there with cloning. There's never been a verified report of human cloning. With stem cells, what we know happened with stem cells was shonky snake oil salesmen popped up all over the world and took advantage of vulnerable patients. Having strong regulation, a demand for a rationale, an efficacy and safety protects patients from being taken advantage of. And we're very fortunate in Australia to have very strong regulations which prevent a lot of tragedies that you see with stem cell tourism. And I think in the gene editing field, if there wasn't the oversight, you would see operators promising impossible cures and taking the money of vulnerable patients. But that won't be happening in Australia, I'm fairly confident.
Okay. Well, I want to come back to that and just expand that a bit later on. But first of all, I want to ask you about your particular research, Merlin. How have you been looking at using these genetic technologies to help people with inherited blood disorders?
So, as Joe mentioned, there are a couple of serious blood disorders called thalassemia and sickle cell anemia. They're very common in people whose ancestors come from the tropics, Mediterranean, Middle East, northern South America. Wherever there was malaria in the past, evolution selected for people with slightly different blood. And if you had one copy of a gene inherited from one parent with slightly different blood, you had a bit of resistance to malaria. But if you had two copies, your blood was not just slightly different, it was defective. And so these diseases, sickle cell anemia and beta thalassemia, are where people have inherited too much of a good thing in a way and have lifelong fragile blood. Now, what you can do is take the blood and the stem cells out of a patient, use CRISPR gene editing to correct the mutation and then get the blood back into the patient. That can represent a cure. As Joe said, that's happening across the world now in various centers. And then this latest work Joe was talking about, which was published only last week or so, where you can actually inject CRISPR into a patient and it can change the blood cells, or this was liver cells. But if you can change those cells inside the patient, then that could be done in many countries in the tropics, and that would be a wonderful thing.
You're listening to Nightlife with Indira Naidoo, and for our Sunday science discussion tonight, we're talking about genome editing technology and all the extraordinary advances that are being made. Our two guests are Dr. Joe Milton, who is an evolutionary biologist and science communicator at the Australian Science Media Centre, and Professor Merlin Crossley, Professor of Molecular Biology at the University of New South Wales, who's working in this area. Merlin, as you mentioned, there've been these amazing advances just recently in this last week. When it comes to being able to administer gene editing treatment directly into the body, how big an advance is that? I mean, you said that it can be now applied to more countries can use this technology. I mean, how big an advance is it?
Look, it's early days, but if this can be done for other diseases, it is a game changer. As Joe said, people previously have done it on things like the retina of the eye, and that's because the retina of the eye is quite small and you don't need to get into that many cells. People have also done this work in mice, but mice are quite small. The attempts to do it in a whole living person have been very frustrating because it's difficult to get enough of this material to change the genes in enough of the cells throughout the whole body of a person. But this new group showed that they did it with lipid nanoparticles, which your listeners will be interested, are the same. It's the same sort of technology that delivers the Pfizer RNA vaccine. Now, those technologies are moving rapidly. If we can use those technology to deliver CRISPR gene editing tools, it's possible we can do a lot more gene therapy on somatic cells than has been possible in the past. And I think that is quite exciting.
It is fascinating, but in a way you're still treating the disease after the person has got sick from it. So I guess the question is, wouldn't it be better to prevent the disease in the first place? Focus on that.
Yes, so that's an absolute key question. It's the right question. So you'd want to do it as early as possible in life, but people are rightfully very cautious about doing medical procedures on children. So the early trials are not being done on children, but if they're safe, you might do it on very young children before they were impacted by these disorders.
Right. When you say very young, what sort of age are you talking about, Merlin?
So with the blood diseases I work on, if you could do it at, before the age of 5, you would avoid many of the lifelong impacts. So I think that would be good. But I think your question is also, would you ever want to do it before a child is born
yeah.
Or conceived? Now, there's an alternate way of going about this, which is with assisted reproductive technologies, where you can have in vitro fertilization, and genetics works in such a way that only 1 out of 4 of the embryos would be affected. So an alternative is just to implant the 3 of the 4 embryos which are unaffected by the mutation. That's an alternative that can work, and I think people will do that in many cases rather than germline CRISPR gene editing.
Okay, this is probably controversial, but is there ever an argument for germline editing?
So it's very perceptive. If you read the documents from the WHO and other august bodies, nobody wants to absolutely rule it out, because if it can be proved to be safe, beneficial, and necessary to help the life of a patient who would otherwise suffer, then there could be some cases. They would be very rare. If 2 people who had sickle cell anemia wanted to start a family, they might want to have the technology to ensure that their child didn't suffer from the same disease as they had, because the genetics would be such that all the children would be condemned to that condition. So it would be a rare opt-in case from particular families. So no one wants to rule it out, but we won't see much of it.
If I can bring Dr. Jo Milton back into our conversation, who is an evolutionary biologist and science communicator at the Australian Science Media Centre. Jo, when people think about genetic modification, as Merlin was mentioning, they do think of babies and what conditions they'll inherit. And of course, we all want to have healthy children. But of course, the danger is that people will start shopping around, won't they, and creating these designer babies. What are some of the concerns you perceive in this?
Well, I think at the moment there are some very valid worries that the technology is simply not advanced enough and accurate enough yet. And we probably also don't really know quite enough about the effects of individual genes to be sure that removing them or replacing them won't cause any problems down the line. Now, one of the big issues with CRISPR is because it targets DNA sequences, it will target the bit you want to edit or remove, but there's also a chance that it could target other areas of the DNA that have the same sequence. And we call these off-target modifications. So we manage to edit the bit we want, but we could be editing unknown other bits of the whole genome while we're at it. So for example, an attempt to alter eye color could also affect other parts of the baby's genetic code, potentially causing health problems. But I think even if it was completely safe, there are still really important ethical questions when it comes to that idea of designer babies, because could we end up creating a kind of 2-tier society? You know, those who can afford access to designer baby technology and those who can't. So the rich could have their children modified to have what could be seen as an unfair advantage over other people if those modifications made them more intelligent, taller, more attractive, that kind of thing. It's a little bit, I think, like the idea of the Eloi and the Morlocks from H.G. Wells' book, The Time Machine. And, you know, in his imagined future, the Eloi live easy lives on the surface of the earth, and the Morlocks live underground, tending machinery and providing food for the Eloi. So I think that's, one of the big concerns. And in terms of that, making sure that particularly medical advances are accessible for everyone and not just the very wealthy, that's, I think, one of the main aims of these WHO guidelines.
And Merlin, these WHO report guidelines have considered a range of fictitious scenarios including, people travelling, medical tourism, going to places which is already happening for dentistry and plastic surgery, cosmetic surgery now, but of course could particularly become a little bit more sinister when people are trying to shop around for a designer baby. What are your particular concerns about where this medical tourism could lead?
So I think, just to reflect on what Joe said, I think the safety of the technology still has to be worked on, but it's rapidly improving, and I'm very confident about that. It's really that people will take money, and there are actually websites already available where people will take money for designer babies, expecting this is possible. But I can tell you, my view is I will never see a designer baby in my lifetime. And the reason is that changing a gene is like changing one road in a city. You can fix a broken bridge, you can fix a pothole, but when you come to want to design a baby, it would be like designing a city. And we all know the city of Canberra was designed, the city of Milton Keynes was designed. It's very hard to design cities. It would be impossible to design a baby. And I can also say, in terms of safety, we only put in genes that are known to exist in normal, in unaffected people. So I think the safety is not the problem. The problem will be that people will be taken in by a dream of designer babies, which is simply implausible and we're not going to see happen.
So where do you think it is mostly going to be applied then, Merlin?
So more of us than anyone realizes carry genetic mutations, and as sequencing of genomes occurs more and more across the population, if you have a relative who has cancer, their genome may be sequenced, or if any of us get cancer, our genomes will be sequenced. And whenever you look into a genome sequence, it's like taking your car to get fixed. The mechanic never comes out and says, oh, your car's looking good, there's no problems there. They'll look into your genome and they'll say, oops, that's going to cause you some trouble down the track. And then you'll think of your kids and think, I wonder if they'll have problems down the track. And some of these problems, the doctors say, these genetic problems, they just get worse. They just watch the patients get worse. In the future, hopefully we can intercede with these genetic disorders that many of us don't know about, and we can stop patients getting worse. I think it is a bright future for humanity, provided it's safe, properly regulated, and people, the profiteers, don't get hold of it. And the World Health Organization and the Australian regulators are trying to ensure that will never happen.
And Jim, back to you. Joe, back to you. We've been talking about genome editing technology because of this WHO report that came out. Can you envisage hearing more from them in this area? I mean, how concerned are they?
Well, yes, I think it's such a hugely promising and rapidly developing area of research and we're really starting to see how useful it can and could be. But it's also going to remain very controversial because, as you sort of touched on at the very beginning, because it's literally tinkering with the stuff of life, if you like. So I suspect the WHO will be watching what's going on around the world very closely and updating these guidelines as required. Now, in terms of their next immediate steps, what they're going to work on right now is putting together this registry for gene editing trials to make sure that there's a good record of everything that's happening. And they're also going to work on this system to help those whistleblowers report bad behavior. And the other thing they want to do is really reach out to the public and help the public understand the science and the potential benefits.
Terrific. Thank you, gentlemen, for joining us tonight on Nightlife. Fascinating discussion. My guests tonight have been Dr. Joe Milton, evolutionary biologist and science communicator at the Australian Science Media Centre, and Professor Merlin Crossley, Professor of Molecular Biology at the University of New South Wales. Thank you.
Thanks, Indira.
Thank you.
And
Thank you.
We've been discussing genome editing technology.
You've been listening to a Nightlife podcast. For more great conversations about the issues that impact you, as well as features on travel and food, head to the Nightlife webpage. You'll find it at abc.net.au/nightlife. You don't need to be a night owl to enjoy the nightlife.